My name is Tatyana Polenova. I'm a professor of chemistry and biochemistry at the University of Delaware. We develop and apply NMR spectroscopy, predominantly solid-state NMR, to study structure and dynamics of large macromolecular assemblies. We're interested in understanding how assemblies in viruses occur and how cytoskeletal protein assemblies work. So we use multiple types of techniques to peer into these kinds of questions with atomic resolution. We studied two major classes of systems. One is viruses, and we work on protein assemblies coming from HIV-1 virus and SARS-CoV-2 virus. So we try to understand how structure and dynamics in the systems relate to their function. Another class of systems that we're working on are cytoskeleton protein assemblies. We look at proteins that bind to microtubules and actin, and we try to understand how their structure relates to their function when they're bound to those cytoskeletal assemblies. A new line of research is we want to understand how proteins are structured and interact with each other and drugs and nucleic acids and so on in the context of living cells. And to do all of that, we use mostly solid-state NMR spectroscopy, but we also use integrative structural biology approaches, including computation, and we couple our data oftentimes with results from cryo-electron microscopy and X-ray crystallography where possible. Our research is based on NMR. Actually, it's a primary tool that we use in our lab, and without NMR spectroscopy, we would not be able to answer questions that we are answering, from fundamental biology to things like how drugs interact with large protein assemblies. And it turns out that for many of those questions, NMR is the only technique that can provide unique information. So for us, it's a tool that's the bread and butter of our research. We're excited in developments of hardware which enable us to do a fast magic angle spinning to look at proteins with increased resolution and also increased sensitivity using very little sample. Ultra-high fields is another advance that will move research not only in our group, of course, but in many other labs forward. And increasingly we're looking at the applications of fluorine-19 NMR spectroscopy to probe structures and dynamics of proteins on their own, in assemblies, in the context of living cells, and the interactions with drugs, inhibitor pharmaceuticals. So that's another area of research that's exciting to us, and we have some new results in this. Another recent set of results actually is to look at structures of proteins which are intrinsically disordered, which bind to microtubules. And if you look at cryo-electron microscopy, it's very little information that is obtainable from that method because the protein is unstructured. Instead, in solid-state NMR, what we saw recently is that when this intrinsically disordered protein, which is called TPX2, binds to the microtubules, we can get its full structure, and not only that, we can explain its function, and its function is stimulating branching of microtubules during the mitosis. So for us it was very exciting to see how the technology instrumentation developments enabled us to look at the system, which is challenging and actually impossible to look at by other techniques. The research into the interactions of inhibitors and drugs with HIV-1 protein assemblies, we expect for it to bear fruition, and based on the results from our group in collaboration with other scientists, better maturation inhibitors will be designed. This is a new class of inhibitors against HIV/AIDS, so this is a direct practical implication of research. So understanding the fundamentals of structures of microtubule-associated protein assemblies will give us insights into things like neurodegenerative disorders, which is another very important problem in biology, both from the fundamental and the applied standpoint. I also want to mention that when we talk about benefits to society, we oftentimes think about very practical applications of what we do. But this is not the only thing why we do science, right? So science gives us fundamental understanding of course, how nature works. It also trains our brain. We work with students, we work with postdocs, and the way we educate them in doing experiments and thinking about the data, it opens a lot of doors for them, not necessarily even in continuing to do NMR, but in all kinds of societal problems and in all kinds of societal activities. So from that standpoint, basic research, including basic research in NMR, all aspects of it, spin physics, instrumentation, applications to biology and chemistry, is really important for society. So we want to use the methods that we developed on isolated proteins and protein assemblies to bring this in the context of the living cells. And you see this direction already is burgeoning in the field of NMR, both a solution and a solid state. And my lab is particularly interested in taking this and seeing whether we can increase the sensitivity to the extent that we can peer at protein structures and dynamics in the context of the living cells.